Welcome to week 8. This week we are going to jump into the first of our chapters on hemolytic anemia. So we have 2 to go over today. So I will make sure I share my screen here. All right, so the first chapter is chapter 23, which is basically an introduction to hemolysis. All right, so the word hemolysis itself means a destruction of red cells. A hemolytic anemia comes about when enough of those red cells have been destroyed and the bone marrow can't keep up. It cannot compensate and replace the destroyed red cells, which then results in the anemia. So anytime you see hemolytic anemia, it's a result of some sort of destruction to those red cells, and the bone marrow just can't keep up anymore. All right, so how does the bone marrow try to compensate for hemolysis? So when there is an increased hemolysis occurring, there's a lack of oxygen, of course, being brought to the tissues since those red cells are getting destroyed. As a result, remember, your kidneys have oxygen-sensing cells in them. They will sense that there's low oxygen in the tissues. They will produce more erythropoietin. That erythropoietin will go to the bone marrow to try to pick up the number of red cells being produced. So that way we can speed up the production to get up more red cells into the blood. So again, if the bone marrow isn't able to keep up, anemia will set in because there's just too few of red cells. All right, so you have acute and chronic types of hemolytic anemia. The difference is the onset. So acute is a rapid onset. So whatever caused these episodes will disappear or subside in between, whereas chronic is over a period of time, at least usually it's generally 6 months or more. They can also be inherited or acquired. Inherited, of course, means you get that at birth. You're passed down by your parents. Acquired means you're picking up something that's causing the hemolysis. Malaria is always a really good example there. If a mosquito bites you and passes you malaria, that would be an acquired process. Intrinsic versus extrinsic, this is involving where it's occurring to destroy the cells. Intrinsic means something about the red cell itself was abnormal. So it's something in the membrane, an enzyme pathway, or something in the hemoglobin molecule inside of it. Something to do with that red cell was abnormal. Extrinsic means outside of the red cells. So it's an external agent that's causing the destruction of the red cells. And then intravascular and extravascular, where is it occurring in the body, essentially? Intravascular means within the bloodstream, so you're having hemolysis within the bloodstream. Extravascular is outside of the bloodstream, typically inside of like macrophages in the spleen and liver. All right, so we're going to start first with learning the extravascular hemolysis pathway, and then we'll dive into the intravascular. So there is a whole pathway process to this. So again, with extravascular, this is involving macrophages, usually in the spleen or liver, so it's outside of the bloodstream. And basically, these macrophages will recognize old red cells and take them out of circulation. So that's kind of their main job anyway. They're looking for different changes to bite out. You know, they're always looking for something abnormal. So whatever is causing these abnormal cells to get extra phagocytized, I guess you could say, by the macrophages, something's causing them to do more eating up of the red cells than normal. So those macrophages will take in the red cell, they will phagocytize them, break them down, and basically what's going to happen is we're going to break this red cell back down to its parts. So when the red cell gets degraded inside the macrophage, that will, of course, lyse it open. Hemoglobin is going to come out of the red cell. And so we're going to break that hemoglobin back down into heme and globin. So it's going to separate back into its 2 parts. Your globin is originally made of amino acid chains, so that's going to go back down into amino acids. So your globin chains will break down into the amino acids they're made of, and those amino acids will float off and be reused in our body for other things. Iron inside of the heme will get released out, and then iron will transport back to the bone marrow to be reused in new red cells. So we're going to reuse our iron. And then all you have left at this point is that protoporphyrin ring. So remember, that's that ring in which the iron was inside the middle before it went off. So we have to get rid of that ring that's left. So what happens is that protoporphyrin ring will react with an enzyme called heme oxygenase and turn into biliverdin. Biliverdin is a green substance. Biliverdin sounds a lot like bilirubin. That's because biliverdin will reduce to bilirubin. Bilirubin is a yellow substance, so it went kind of from a green to a yellow substance. And so a lot of times if you've been in clinical chemistry or just kind of generally heard about bilirubin, you may have heard it referenced as the waste product of hemoglobin, and this is why. Truly, when you break down your red cells and break back down your hemoglobin, it eventually will turn into bilirubin. So at all times in your body, we are getting rid of old red cells. It's going through this exact process. So we're always having a little bit of bilirubin at any time in our body. When you pick up and have a hemolysis occurring where you're breaking down extra red cells more than you should, then you will see an increase in bilirubin presence. So that bilirubin is always an increased level in any type of hemolysis. So once we get the bilirubin, that's going to join up with albumin and go to the liver cells. In the liver, it's going to join with 2 glucuronic acids to make what we call bilirubin diglucuronide, or in the world of lab, we call it conjugated bilirubin. And that is actually a test that we have in the chemistry world. So that conjugated bilirubin is going to get excreted out of the liver through the bile. The bile goes to the intestines, and then that conjugated bilirubin will turn into urobilinogen. And just like the first part of that urobilinogen sounds, uro for urine, we then excrete the urobilinogen into the urine, and there's your process. So we have completely taken a whole red cell and broken it back down to its absolute parts and done away with it. We've reused what we could reuse, and then we've gotten rid of what we need to get rid of. So that is the hemolysis process overall. So that's extravascular. Now, that's anything that's already starting outside of the bloodstream in the spleen, the liver, or just in tissues in general with macrophages. So keep this in mind. I do need you to know these steps in kind of that order there. Now, there is an intravascular hemolysis process. So what this means is instead of already starting in the macrophages and the tissues, we're starting inside the bloodstream first. It's intravascular, so within the bloodstream. So here, something is destroying our red cells, and those red cells will lyse in the blood and release out hemoglobin. So you'll have hemoglobin floating in your blood. So that's what happens here first. Because hemoglobin is a smaller molecule, smaller, whatever you want to call it, smaller protein, it can actually fit through the kidney glomerulus filtering system, and we don't want that to happen. We want to try to save the iron inside the hemoglobin. We want to try to save those amino acids and just basically reuse what we can. So we're trying to save it. We don't want all of it to get filtered out. So we have 3 ways that we can try to save this hemoglobin and the iron and amino acids in it from being lost through the kidneys. We use haptoglobin, Hemopexin, and albumin. So those 3 proteins are going to become involved here. So haptoglobin is just like the name sounds, haptoglobin. It sounds really close to hemoglobin. The entire role of haptoglobin is it binds hemoglobin and carries it to the liver. So that will prevent it from being filtered to the glomerulus, and it will keep it and bring it to the liver, and so then it will undergo that exact process that we just discussed in the extravascular in the liver. So it will break its parts back into amino acids, the iron will get reused, and we'll break down that protoporphyrin ring into biliverdin, bilirubin, conjugated urobilinogen, and so on. So that whole process that we just discussed will happen. We just got to get it there. So the haptoglobin is really good about binding the hemoglobin and transporting it to the liver to do that. That's its job. So this is just kind of going through those steps. Now, the one key note here is that very last line on the slide. When haptoglobin does this job, it's depleted in the process. So we're not going to be able to reuse the haptoglobin. It kind of does the job and it's depleted as a part of it. So just know that you will see decreased levels of haptoglobin anytime there is an intravascular hemolysis process happening. And that's simply because it's doing this job of bringing the hemoglobin to the liver, but it's depleted. It can't come back from that. So it's a low level you will see with that hemolytic process. All right, another protein is hemopexin. Same thing. It's going to bind with the hemoglobin to save it from the kidney filtering. When it binds with that hemoglobin, it basically is going to, you can ignore the whole second stage there. Basically just know it binds with hemopexin to save it from the kidneys. Then they will bring that to the liver just like the haptoglobin. Hemopexin is able to be recycled though. Once it brings it to the liver, drops it off, it goes back. It can be reused. We can keep using hemopexin. And then the third one was albumin, or what you can call methemealbumin. So albumin is going to bind with methemoglobin. So the hemoglobin will have purposely changed its iron state into that ferric Aq 3+ state, where that makes that methemoglobin. We've talked about that in chapter 10. So again, methemoglobin, anytime your iron is in that ferric 3+ state instead. So it will purposely change its iron and make itself methemoglobin. So when the albumin binds with that, it forms methemealbumin. But again, the role is to save it, to prevent it from being filtered by the kidneys. So once hemopexin is freed up, it will release it off the albumin, it will join up with the hemopexin, and then that again transfers it to the liver. So again, all three of these proteins, goal is to save the hemoglobin from being filtered as much as we can and bring it to the liver so that we can break it down just like we learned in the beginning process. So again, that extravascular hemolysis process, all those steps are really big to know because that's the majority of what it's going through. All right, so excessive extravascular hemolysis, this is kind of, this really isn't that important of a slide to me. Really, I could almost delete this and we'd be fine. It's just discussing that most of the time when our red cells get older, they kind of have these markers on the surface to tell the macrophages, hey, I'm kind of old, I'm ready to be done. But in certain disease processes, they will put those markers on the red cell surface even though they're not that old, and that's why it will result in excessive hemolysis occurring. So it's just kind of informational. This slide isn't that necessary for you. All right, so I'm actually going to come back to that. We're not going to worry about that last mention there. I might come back to it here. We're going to hang on to the last slide for a second. So overall, in this chapter 23, the introduction, the key is knowing that hemolysis process. Why is this? Okay. So again, if it starts in the intravascular pathway, it means it started within the bloodstream. So I want to list you some key signs of what it means to have intravascular hemolysis occurring. I thought this would make it bigger. No, it's not. Okay. I was hoping that would make my writing bigger. All right, so a lot of signs of intravascular hemolysis are schistocytes. Remember, those are your fragmented red cells. And so if you have an intravascular hemolysis occurring, we're going to see schistocytes, fragmented pieces of red cells, which should make sense. If your red cells are getting destroyed in the blood, and then we go look at a peripheral blood smear, you should see those destroyed red cells, those little pieces floating around. So schistocytes are always a really good sign of an intravascular process. Another sign would be keratocytes. Remember, those are the helmet cells. Keratocytes are found in all the same conditions as schistocytes. So they're both found together. They're both big signs of intravascular hemolysis occurring. So that's sign number 2. The 3 hallmark signs of an intravascular hemolysis is, one is hemoglobinuria. This means that you have free-floating hemoglobin in the blood. So if we think about it, when your red cells get destroyed in the blood, you're releasing hemoglobin right out into your blood. So of course, you're going to see free-floating hemoglobin in the blood in intravascular hemolysis. Another sign is going to be hemoglobinuria, which means you have hemoglobin in your urine. So we discussed having those 3 proteins, haptoglobin, hemopexin, and albumin, to save the hemoglobin and prevent it from being filtered by the kidneys. However, you can't prevent it all. It's going to do its best job to save what it can, but there always is going to be some hemoglobin that gets through the kidneys into the urine. So as a result, you will see it in the urine. And then finally, you have hemosideruria. Remember, hemosiderin is a form of stored iron. Well, if you think about this, hemosiderin, I can't talk and spell at the same time, hemosiderin, which is a form of stored iron in urine. Okay. So anytime that you had hemoglobin being filtered, iron being filtered out, you're going to see iron in the urine as well. So these things are getting through the kidney filters, even though we're trying our best to save the stuff. So those are 5 really key signs of an intravascular hemolysis occurring. You will not see these 5 items in an extravascular. These are unique to just the intravascular inside the bloodstream only. Because in the extravascular, you're not in the bloodstream. You're not being filtered by the kidneys. You're over in the spleen, the liver, and the tissues. So it's not going through this process. So these are very unique to just that intravascular process only. So I wanted to make sure I mentioned that. All right, so again, your key proteins are haptoglobin, hemopexin, and albumin that will try to bind that hemoglobin and bring to the liver. And once in the liver, it will go through the exact same process as the extravascular hemolysis. So again, remember, the only one out of this, haptoglobin, will be decreased because it does not get reused. We cannot bring it back. Once it does its job of delivering the hemoglobin, it is depleted. So you always see a decreased haptoglobin as part of an intravascular process. All right, so with that being said, I want to just review the extravascular pathway again. I'm just going to put it into quick, simple terms. You know, be sure to go through, you can re-look at the slides. I know I don't read them off per word, but you can read them yourself. You can re-listen to this lecture, whatever you need to do, but I just want to quick list out the steps in general. So again, your red cell is going to get phagocytized by the macrophage. So that macrophage could be located in a variety of places, but at any rate, it's going to get phagocytized. When it does so, your hemoglobin is going to be broken back down into its parts, heme and globin. Remember, your globin is made of amino acid chains, so your globin will break down back into amino acids, and we are going to reuse those amino acids. So they'll go off and join the amino acid pool in your body. Your heme will release the iron, and that will bind with transferrin and go to the bone marrow to get put back into new red cells to be reused. So we're going to reuse our iron. And then finally, we have that protoporphyrin ring left of the heme. So that protoporphyrin ring will reduce down to biliverdin, which again is a green substance. That will change into bilirubin, which is a yellow substance. If this is not happening in the liver, that bilirubin will join with albumin and go to liver. If it's already in the liver, it doesn't need to go there. It's already there. Once in the liver, bilirubin will join up with 2 glucuronic acids, I can't spell, and make conjugated bilirubin. That conjugated bilirubin will be excreted in the bile to the intestines. Once in the intestines, it will convert to urobilinogen. And again, urobilinogen, like the name implies for uro, is excreted in the urine. So that's kind of a quick overview of the process of extravascular. So no matter where it starts, whether it starts within the bloodstream or outside the bloodstream, at some point it will go through those steps of the extravascular. So very important that you understand those steps. Now keep in mind some things that will be seen here. With both intra and extra, both are going to have increased bilirubin levels. So both intravascular and extravascular have increased bilirubin. And again, that's because most of us are always breaking down a red cell that's old. That's a normal process. And so that's what it's known as a waste product of hemoglobin. At some point, it will always change to bilirubin. Now, whenever you have hemolysis, an increase in that breakdown process, you're going to get increased bilirubin eventually. So increased bilirubin, always a sign of some sort of hemolysis going on. So you will see that. As a result of the increased bilirubin, you're also going to get increased urobilinogen because it's going to convert to urobilinogen. So you will always see increased urobilinogen levels as well as a result. So just kind of an FYI on that. So, all right, we'll just kind of leave that as is. So very important to know these pathways and know some of these key signs and how to separate between an intravascular and an extravascular process, especially by those key signs that we listed up in the top under the intravascular. All right. One thing I should mention, I keep thinking I want to mention it in this chapter. One unique cell to the extravascular are spherocytes. So spherocytes are those very round red cells with no central pallor. They're very compact, they're smaller, but they have no central pallor. And so the reason that spherocytes can be a sign of extravascular hemolysis is that when your blood goes through the spleen, you have macrophages in the spleen looking for things that shouldn't be there. And what happens to form spherocytes in extravascular hemolysis is your macrophage will go to take a bite out of the red cell, like maybe remove an inclusion or phagocytize that red cell, and it doesn't get the whole thing. It only bites a portion of the membrane. What happens then is it took a portion of that membrane, so the spherocyte seals its membrane back up, and as a result, it has less surface because part of its membrane is missing and it had to seal itself back up, but it still has just as much stuff on the inside. So it's like a hard, compact ball. It's very inflexible. It's not going to be able to maneuver through those small spaces anymore because it's so compact, filled with stuff on the inside, but a very small surface area. And so as it's trying to get through the small areas, it tends to burst easily, leading to more hemolysis. So spherocytes, always a really good sign of extravascular because of that. I do ask that you get familiar with how to list out that happening, getting familiar with, if I ask you on a test to describe how a spherocyte is formed, be able to list out that when it travels through the spleen, a macrophage will take a portion of the membrane. It doesn't get the whole cell for whatever reason, maybe just gets an inclusion out, and the cell reseals itself, has a smaller surface, but it's so full of the normal stuff on the inside that it turns into this compact, hard, rigid ball, and it can't get through those spaces anymore and bursts. So that's one way that a spherocyte is formed, and it's very much a sign of extravascular. It's not a sign of intravascular because it's happening in the spleen. So keep that in mind as well. All right, so I think that was a lot of information in the first chapter alone. All right, let's skip over to our next chapter, which is chapter 24. All right, so chapter 24, quite the title, Intracorpuscular Defects Leading to Increased Erythrocyte Destruction. So let's break this down. Intracorpuscular defects. This is meaning that something inside the red cell is defective. So something about that red cell is defective, which leads to hemolysis. So that's what we have happening here. So all of these disorders in this chapter are going to be a red cell defect, and it results in hemolysis. So we're going to start first with those defects of the red cell membrane. These are hereditary, so you're born with them. Two major ones in here are hereditary spherocytosis and hereditary elliptocytosis. There is a third one called hereditary pyropoikilocytosis. So we'll go through each one. First up is that hereditary spherocytosis. So as the name implies, you're going to see a lot of spherocytes in the blood on this one. It is a hemolytic anemia because, again, spherocytes are very compact, rigid little balls that can't get through small spaces and will easily burst. So that's the hemolysis part. That's what results in the hemolytic anemia. So the spleen is the #1 cause of that hemolysis, which results in the anemia. Because it can't transverse those small spaces in the spleen, that's what's causing the hemolysis. So hallmark signs of this are increased fragility, which means the red cell is fragile. It can't get through anything. It's easy to burst. That's what it means by fragility. It's easy to burst. And then reticulocytosis. Anytime we have hemolysis occurring, your bone marrow is working extra hard to get out any sort of red cells it can. So usually what happens is it will kick out a ton of reticulocytes just trying to get any sort of red cells out there. So you will always see increased reticulocytes anytime we have a hemolysis occurring. All right, so in hereditary spherocytosis, how these form is there is a defect in the membrane proteins, and it's specifically in spectrin, anachiron, protein 4.2, and BAM3. You don't have to remember all of those, but I would remember that it is a defect in the membrane proteins. And so as a result of this defect, it basically causes that red cell membrane to lose unsupported lipids. So this membrane is essentially peeling off the red cell. So it's still, again, containing all that volume on the inside. It's just the membrane peeling off. It's resealing itself. So again, it has that decreased surface area, but high amount of volume on the inside to make it a spherocyte. So really, at the end of the day, there are two ways to really make a spherocyte. One is the way that we just discussed in the extravascular pathway, where the membrane takes a, or the macrophage takes a bite of the membrane and then it seals itself. This is the other way in a hereditary condition. This is only seen in a hereditary condition to form it this way. So no matter what, both of them are going to form spherocytes, which are, again, are rigid and can't get through that spleen. So we will see, of course, anemia. You're going to see jaundice because it is a hemolysis. It's a hemolytic process, so you're going to see increased bilirubin levels, which lead to jaundice, yellowing of the skin and eyes. You're going to see splenomegaly, which is an enlarged spleen. Because it's all about the spleen in this case, with the spherocytes getting stuck down there, it's going to cause, you know, kind of an enlargement of that spleen. It's going to really, can't think of the word I was trying to think. But at any rate, it's going to cause enlargement of the spleen because that's where it's happening and they're getting stuck and it's, you know, causing issues in that spleen. As far as the lab goes, on the microscope, you should definitely see tons of spherocytes. In the CBC, this is one of the few times we're going to see an elevated MCHC, that hyperchromic. We don't love to use that word, but this is probably the only time it's okay to say that word when you see a ton of spherocytes. So normally that MCHC should never really be high. It should always be in the normal reference range or lower, but this is one of the few times it can be high. So a high MCHC is a very key finding with spherocytes. There is what we call an osmotic fragility test that will very much be positive here. So that's basically saying, again, your red cells are extra fragile, they're extra bursting. Again, you have high reticulocyte counts because it's a hemolysis. We're trying to replace those destroyed cells. In your bone marrow, if you were to see your bone marrow, you're going to see erythroid hyperplasia. What that means is that you can tell that they've really increased the red cell production. There's a lot of red cells getting produced. So we're going to leave it at that. Ignore the last line. I should actually cross that off. Don't pay attention to the last line there. All right, so here's what a smear would look like with spherocytes. So as you look around, you can see a lot of those cells have no central pallor. They're smaller in size. You know, compare them to that lymphocyte nucleus that is showing there. You can see most of them are smaller than that and have no central pallor. That's a key sign of spherocytes. So here's a little bit on that osmotic fragility test. What they do is they will place red cells into a solution. Water will enter the cell until equilibrium is established. When you have a spherocyte, because they have so much content on the inside and less surface, they can't handle very much water. So when you try to put water into them, they will quickly burst. So they're increased fragility, whereas a normal red cell can handle quite a bit of water for a while. So spherocytes will burst faster. That's kind of the key end result of osmotic fragility. There is another test called autohemolysis. I am not going to test you on this. You can ignore this one. It's not really used anymore at all, so just ignore it. So treatments, ideally the treatment is splenectomy. Remove the spleen because that's what's causing the hemolysis. That's where the cells are getting stuck and bursting. So if you can function overall and get cells that aren't bursting all the time, you should be rid of your anemia for the most part. So by removing that spleen, it's going to help solve it. They will always have spherocytes. It's a hereditary condition, but it will keep it from being so significant. All right, on to our next one is hereditary elliptocytosis. So again, this is a defect in the red cell membrane skeletal proteins. In this case, it is spectrin, especially that is abnormal. So it will basically turn that shape into that elliptocyte shape, that elongated oval shape is what they will look like. So in the lab, in the smear, you're going to see definitely tons of elliptocytes, lots and lots. Now, you can see elliptocytes in quite a few other diseases like iron deficiency anemia, thalassemia, but when it's hereditary elliptocytosis, you need more than, I think, 30% of their red cells to be elliptocyte in shape. That's a lot. Essentially, you need to see a buttload of it. So more than 30% of the red cells should be in elliptocyte shape in order to be this. They will have an anemia. That shape doesn't always make it through everything. Again, it can get stuck. Splenomegaly, because that's kind of where it's getting stuck, and then resulting in hemolysis. So there is what the elliptocyte shape looks like. You can see that one, I know it's a zoomed in field, but almost everything there is oval in shape. All right, hereditary pyropoikilocytosis. Oh, that's a mouthful. Basically, as the name implies, tons of different shapes, odd shapes even, and then the pyro part is referring to heat. So we're going to kind of get into that here, but it's a severe hemolytic anemia with extreme shapes, extreme poikilocytosis. So you're going to see elliptocytes, red cell fragments, budding red cells, spherocytes, lots of different things can show up there. Splenectomy is the ideal treatment so that these cells don't get stuck in the spleen. I'm not going to worry about the other features there. But when you apply heat to the sample, so the top part is normal, what they look like on its own, lots of poikilocytes in the top one, and then when you take that sample and apply heat to it, you can see just how much more misshapen those cells become, and that's what the name pyropoikilocytosis comes from. All right, so all I would remember on this one is that it's lots of varying poikilocytes with budding red cells, pencil-shaped red cells, elliptocytes, that kind of stuff. It's a lot of varying types of poikilocytes. All right, on to some inherited disorders that involve your sodium and potassium kind of pumps. You know, we're always pumping potassium or sodium in or out. So you can actually have a defect in that, in the hydration level of your cell. Basically, you can have cells that become stomatocytes that are overly hydrated, that have water coming in too much. Or you can have the opposite, where you have dehydrated cells known as a xerocyte. So hereditary stomatocytosis is when you have overly hydrated cells. You have too much water coming into the cell, creating the stomatocyte shape. So again, that's where the central pallor is shaped as a rectangular, or like a slit, if you will. So you're going to see a lot of stomatocytes. Again, most of the cells should look really stomatocyte in nature. So there it is, that rectangular kind of central pallor is the key sign of a stomatocyte. That's all I really need there. I don't test you a ton on this at all. And then hereditary xerocytosis is dehydrated cells. I will not test you on this. You can ignore it if you like. All right, moving on to hereditary acanthocytosis. So anytime an acanthocyte is formed, it's usually due to something about the lipids in the membrane are abnormal. So in this case, with the hereditary condition, they have abnormal membrane lipids, specifically sphingomyelin, but a decrease in others. So the shape will become this acanthocyte shape, which is these uneven projections is essentially what an acanthocyte is. Again, most of your cells are going to be that acanthocyte shape, very much a hemolytic anemia. Also pretty much can be associated with what you see there, liver disease, A-beta lipoproteinemia, that's always a fun word. Things that involve lipids. Your liver makes a lot of your lipids. A-beta lipoproteinemia is meaning that you're without certain types of lipids, so things like that. All right, oh good, this is on here. So red cell enzyme deficiencies. There is a big one called the G6PD deficiency. That enzyme is glucose 6-phosphate dehydrogenase, and maybe you've heard of it from clinical chemistry, but that enzyme is essential in our hexose monophosphate pathway. So, right, I don't know if you can see my mouse, but hexose monophosphate shunt. You can call it a pathway if you want. Remember, we learned about this pathway back in chapter 9. This pathway's whole job was to give us glutathione, and glutathione's job is to protect our red cells from oxidation. So that's what it does. If you are missing or deficient in that enzyme G6PD, which that means you can't go through the hexose monophosphate pathway. It won't work because you're missing that enzyme that's part of it. As a result, you're not going to make glutathione, which means you're not protecting your red cells from oxidation. So your red cells are very vulnerable to oxidative damage. So if you were to take certain medicines, certain drugs that are prone to that, then it would result in hemolytic process for you. You might not have hemolysis occurring at all times, because again, it's only when you are exposed to oxidative damage. So maybe if you took a certain drug or something like that, then it might happen. So again, it's all about missing this enzyme and not being able to form glutathione to protect our cells. So very, very key finding in G6PD deficiency is Heinz bodies. Remember, Heinz bodies are precipitated hemoglobin. So that is a very key finding in G6PD deficiency. I would always remember that. So you'll get that precipitated hemoglobin, those little Heinz bodies in there, and then when they go through the spleen, the macrophages will see them sitting in the cell, and again, the macrophage's job is to get rid of it. It's abnormal. So it gets rid of it. Well, that's part of the hemolysis. Now you're hemolyzing your own cells because you're seeing these little chunks of hemoglobin there. So of course, it's a hemolytic process, so you're going to see increased reticulocytes. A lot of times this might be happening. The hemolysis could also be happening with the oxidative agents inside the bloodstream, making it more of an intravascular process. So remember those Heinz bodies. And remember, Heinz bodies are the ones that can only be seen with supravital stain. You're not going to be able to see them under normal right stain on a peripheral smear. You would have to stain it with supravital in order to see the Heinz bodies. Another enzyme deficiency is pyruvate kinase deficiency. This is probably the more common one. This is an enzyme that's in the glycolytic pathway. It also results in a hemolytic anemia. So you can see pyruvate kinase deficiency with, you will see burr cells in there, which we call echinocytes. So those are kind of a poikilocyte that you might see with that. And then we have paroxysmal nocturnal hemoglobinuria, otherwise known as PNH. This is probably the one that causes people the most problems on test questions. PNH is a bugger for some people on test questions. So I'm just going to warn you of that. It is a hemolytic anemia. In fact, it's an intravascular hemolytic anemia. So it's occurring within the bloodstream. So you're going to see those signs of an intravascular hemolysis with the hemoglobinemia, hemoglobinuria, hemosideruria. Those classic intravascular signs will be seen here as a result. This is the only one that is not hereditary out of this chapter. This is acquired. So important that you write that down. So in this chapter, we've pretty much gone through just hereditary conditions. This one is acquired. It is an acquired red cell membrane defect. So you're not born with it, you picked it up. So that red cell membrane will become defective somewhere in your lifetime. As a result, it really increases your susceptibility to complement. So if you haven't been in immunology yet, or if you're in it and you hate it, whatever it might be, complement is a series of proteins that are part of your immune system, and they're meant to destroy. They will target something that's foreign to them, and their goal is to destroy it. Let's leave it at that. If they destroy it, well, in this case, if they are seeing that defective membrane of that red cell as something they shouldn't be there, they will set out and destroy it. By destroying it, that results in your hemolysis. So very important you remember, it's intravascular hemolysis. It's due to an acquired red cell membrane defect, very susceptible to complement destruction, which is what's giving it the hemolysis occurring in the disease. Now, the reason they named it nocturnal hemoglobinuria is because about 25% of people, when they would wake up, they would have hemoglobinuria. They would have basically red urine. So hemoglobin in their urine, red urine after coming from sleeping. So it kind of occurred overnight. That's where it got the nocturnal name. So again, intravascular process. In the lab, we're going to see pancytopenia, which means all three cell counts are going to be decreased. So decreased red count, decreased white count, decreased platelets. It is a normochromic normocytic anemia, and actually all hemolytic anemias are normocytic, normochromic. There's nothing wrong with the red cell size for the most part, unless they're like spherocytes. But overall, the red cells tend to stay the same size if they're normal. So for the most part, hemolytic anemias will fit in that normal category. Of course, all hemolysis, including PNH, will show an increased reticulocyte count. So again, and then you're going to see those classic findings of an intravascular process, the hemosideruria, hemoglobinuria, and hemoglobinemia. So all three are very much signs of an intravascular hemolysis. So once you kind of see all that and you have a thought, there's some screening tests that can be done that the doctor can order. The first is called a sugar water, or you can call it sucrose hemolysis, so sucrose for sugar test. If that is positive, then there is this Heme test that they use to confirm. Now, I've heard a lot that they don't use that test very much anymore. It's kind of gone by the wayside. But I do want you to know those tests because your board exam was still asking questions on it. So as a result, I want to make sure we're always prepared as best as we can be. So sugar water sucrose hemolysis is the screening. If that's positive, the Heme test, otherwise known as a serum lysis test, will be used to confirm it. So those are the two key tests to help diagnose P and H. Okay, so that's it. Lots and lots and lots of information in these two chapters. Really listen to it, take notes, go through it, you know, get hands-on. It's doable, you guys. It's doable. So if you guys have questions, as always, please feel free to reach out at any point and have a fabulous week. Thank you.